Hello, this is the poster for exploring the insulin resistance molecular landscape with knowledge graph embeddings. Biomedical knowledge graphs aid drug target discovery by organizing entities and relationships from various biomedical data sources and incorporate diverse data types, such as protein interactions, metabolic pathways, gene expression, and disease associations as a set of nodes and edges. Knowledge graph embeddings are vector representations of the entities and relationships in a knowledge graph and can be created using various kinds of graph data science algorithms. The embeddings can be used as input features for traditional machine learning and graph neural network models to gain a better understanding of disease biology, identify new drug targets, and aid target prioritization for experimental validation. Several approaches exist for discovering novel gene disease associations using knowledge graph and knowledge graph embeddings. However, disease-agnostic identification approaches of novel drug target for complex phenotypes, which involve several diseases, comorbidities, and biological functions, are still underexplored. Insulin resistance is a particularly complex pathphenotype, which impacts several organs like muscle, brain, liver, even gut, under a variety of environmental and genetic conditions. Insulin sensitivity plays such a central role in the regulation of metabolic processes that the identification of drug targets for its regulation is a complex task. We use knowledge graph embeddings and features engineered based on knowledge graph topological properties for exploring the pathophenotype of insulin resistance and identifying drug target candidates. Our hypothesis is that knowledge graph embeddings and knowledge graph topological metrics capture biologically relevant information that can be used with machine learning models and graph data science algorithms to distinguish genes that are related to insulin resistance from those that are not. Our approach is being developed and tested onto the public biomedical knowledge graph called OpenBioLink. We use two positive sets of different sizes. The first one has 624 genes, and it has the genes identified by Gao et al. In their publication Underlying Mechanisms of Insulin Resistance, a bioinformatics analysis based on validated related genes from public disease databases. The second one has 55 genes, which are annotated as related to insulin resistance in the Human Phenotype Ontology. We employ the following three approaches. First is a supervised machine learning classification ensemble model using topology-based features. The second one is doing lean prediction using translation-based and graph neural network-based knowledge graph embeddings. And the third one is doing positively unlabeled learning approaches. We tested El Canoto, Locaro layer factor, isolation forest, and one-class SVM, using as input the knowledge graph embeddings calculated in approach number 2. We evaluated our models with the confusion matrix, area under the ROC curve, mean rank, mean reciprocal rank, adjusted mean rank index, and looking at the top 10 and top 100. In addition, we characterize the biological relevance of our gene predictions based on three different approaches. First, we use the diffusion profiles from the orthogonal knowledge graph called multiscale interactome, which identify the paths connecting the drug and the disease that have potential relevance for the drug mode of action. We calculate the percentage of the top 100 scoring genes that have a statistically significant weight in a diffusion profile from a drug for an insulin-resistant related disease. Then, using the depth map correlation-based clusters, we have a second approach that looks at the functional annotation of the clusters. So the clusters containing the insulin-resistant related genes from our positive set were identified, and then we identify which genes from our top 100 predicted genes map to any of these clusters. As a third approach, to automate the literature search for each of the top 100 genes, we implement a text mining plus GPT approach that extracts all the abstracts from PubMed, matching the search gene, insulin resistance, and then performs Q&A on that corpus. We found that Rotate-E outperforms every tested method in the large training set, and El Canoto on the Rotate-E embeddings is the best performing one on the other scenarios. Interestingly, with a large training set, even the simplest method of machine learning model with topological features is the second best performing approach. Notably, We also observed that knowledge graph embeddings from insulin-resistant related genes lie close together in some pockets in the embedding space, rather than being all spread randomly all across the space, suggesting that indeed the knowledge graph embeddings capture the biological information that makes them similar in the sense of being part of the same complex biology, while genes not related to insulin resistance lie outside of these insulin-resistant related neighborhoods. In summary, by utilizing a variety of graph data science, machine learning, and positive on-label algorithms, we aim to provide a workflow for identifying potential drug targets for insulin resistance that can also be easily extended to other complex phenotypes for disease-agnostic drug target identification approaches. Thank you very much for your attention.